Hey everyone, and welcome to Chapter 16, Part A, which we will use adenine for A today, where we're going to talk about the evidence leading up to us understanding DNA as the heritable material and the evidence and the story behind how we came to understand DNA having this double helix structure. These final two chapters, chapters 16 and 17, we're going to be getting at the mechanics for the central dogma, if you will. That is to say, going from DNA to messenger RNA to proteins, getting us to these macromolecules that then allow us to do things like make structures or undergo functions or store energy in some cases. All of the different molecules out here are derived from the central dogma right here. And so understanding DNA and its chemistry opens a door for so many things in terms of biology and medicine. It's hard to overestimate the importance, and that's why we spend time on it. Just an example of something that you've probably encountered in everyday life is all of the explosion of companies that will analyze your DNA and tell you about yourself, your heritage, and or risk factors for certain types of diseases. 23andMe is probably the most popular site, in conjunction with Ancestry.com. But we are rapidly evolving an understanding of the connection between the genetic code and the risk for diseases. And I say risk because it's very important to discriminate between what will actually happen and what could happen. Risk gives you probability, and that's the truth when it comes to our genetic material. That yes, you might have an increased likelihood or probability, that doesn't make it an absolute certainty. But these companies are making literally billions of dollars now, not only in the commercial markets where we are doing things to understand our history and that sort of a thing, but also in the medical markets and criminology and so forth. So we can diagnose disease based on our understanding of the chemistry of DNA, or things that may have triggered a disease, or how severe the disease will be, and what medicine can be used. There's actually an effort going on to get 1 million people to have their DNA examined so that we can identify genetic markers that may say, hey, this might be a better therapy than another. One of the goals with this chapter is for you to understand the structure and function of DNA beyond what you already know for sure, and understand the experimental evidence. That is to say, you need to know the major contributors to the current understanding, the major experiments, how we came to conclude that DNA is the heritable material, but most importantly, be able to interpret experimental evidence. So I may give you some evidence and then ask you to interpret what the meaning of that evidence is. Rather than going lockstep into here's, DNA is a heritable material and that's the end of the story. We're going to look into how we came to solve that problem, because we didn't really know for a long, long time until relatively recently that DNA was the key material. A big part of understanding DNA was getting to understand its structure. Its structure was not immediately evident. In fact, it was a great race by several researchers around the world, by literally dozens and dozens of researchers around the world, to figure out what the structure was, because we can't see it under a microscope. Not even hardly an electron microscope can we see it. Maybe because part of the reason why is that electron microscopes are so powerful in terms of the energy that they transmit that the energy to observe these molecules destroys them in the process. We're able to use X-rays, but electron microscopy is very, very difficult to see DNA strands, even with modern techniques. And so there was a race to figure out the structure. And in 1953, the famous Watson and Crick. James Watson, who did attend Indiana University for a period of time, as a graduate student, is one of the two that helped postulate the structure that has given rise to our understanding of what it is today. It is the most celebrated molecule of our time because the understanding the code, understanding the behavior has given us so much insight into the functioning of cells and in disease. So we know, as far as we know, all life on this planet requires DNA at some point. Even viruses that are RNA viruses, like for instance COVID-19, needs DNA because the virus can't replicate itself. It needs to have assistance from a host cell, and that host cell has DNA. So all life as we know it depends on DNA. And it is the storage, and it's the storage molecule for information. In fact, commercial companies are actually looking at DNA as in terms of artificial storage now. The efficiency with which information can be encoded in the DNA is actually orders of magnitude beyond the best algorithms we have for compressing information on digital media at this point. So they're actually working on DNA-based information storage. If you ever get a chance to go over to the Indiana University School of Medicine in the Van Nuys Medical Science Building, an artist by the name of Dale Chihuly, who's a famous glass artist, constructed a huge molecule of DNA using the atomic radii of the different atoms involved in DNA. And it's about a 20-foot tall structure that you can see in the science building there. But the original structure was made out of Tinker Toys. And here's James Watson and Francis Crick, and they're standing around looking at their molecular model, because when they proposed that structure, it had to work in terms of the data that we had about the molecule, and we did have some data about how wide the molecule was and how long the bond angle or the bond angles and so forth. And so here they are. So it had to work on a physical level in addition to a conceptual one. And here is the Nobel Prize winning paper. If you go and look up a research paper nowadays, most of them would never be about 10 paragraphs long. In fact, the best paragraph of all is right here. It says, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism." So they were able to describe the structure of DNA, and then that's probably the understatement of the century right there in that one paragraph. So they were awarded a Nobel Prize for their postulation of what turned out to be the correct DNA sequence structure. Let's break into the evidence now. It actually was only a few decades earlier that we even thought DNA had a true functional role in cells. We knew that cells had to be able to pass genes or traits on to subsequent generations, but it was mostly theorized that it had to be carbohydrates because they had the biggest diversity, or proteins or something. In fact, DNA was so repetitive that they thought maybe it was just chaff, like it was just, redundant stuff in the cells. That all changed in the 1920s with an experiment by a gentleman by the name of Frederick Griffith. Griffith showed that DNA could specifically change the heritable traits in bacteria. So he had two strains of bacteria, one that was lethal and one that was harmless, one that could be given to animals and not cause disease. The other one did cause disease and death. What he found was that he could kill the disease-causing strain of bacteria, mix it with the harmless bacteria, and then those harmless bacteria became pathogenic. So what was otherwise a harmless strain, when mixed with the remains of pathogenic bacteria, could themselves become pathogenic. That term of changing the phenotype of an organism is called transformation. When we transform a cell or transform bacteria, what we're doing is we are changing the DNA structure inside that organism. We are adding or subtracting sequences of DNA to their chromosomes. That changes the genotype and thus changes the phenotype. Phenotype is the manifestation of traits and genes. So he was able to bust up the pathogenic, incubate the remains, and the new bacteria became pathogenic. So let's look at how the experiment went down. So we had to have 3 basic experiments before we started. We had to be sure that the S cells that we were using were in fact pathogenic. So when you inject them into the mouse dies. That would be called a positive control for pathogenicity. Then you have to have a negative control, right? So we inject the R non-pathogenic bacteria into the mice, and the mice were just fine. Now, this is listed as a control, but I would tell you that it's actually more than that. It's probably a partial experiment. When we kill those S cells with heat, we know where we find that those mice do not get sick and die. So we have the positive control has been experimented with heat, and we have inactivated those. But when we mix the heat killed and the R cells, the mouse not only dies, but when we look at the bacteria that we injected, the bacteria have the phenotype of the S cells. Even though we didn't inject living S cells here, we now find living S strain cells in that dead mouse from the mixture. So something from the heritable material of these S cells was transferred to the R cells and transformed the R cells into S. Knowing that there was heritable material that was non-living in the cells was 1928. It wasn't for another 16 years that Avery, McCarty, and MacLeod were able to proposed that the transforming substance found in those heat-killed cells was in fact DNA. They proposed it. We're not going to get into the experiment, but the long and short of it is that they were able to chemically separate the different molecules from those heat-killed cells. They were able to extract DNA and extract lipids and extract proteins. And the only one that worked in transforming the harmless bacteria into pathogenics was the extracted DNA. That said, because we didn't know a lot about the structure of DNA, many folks in the scientific community remained skeptical. They were like, oh, it's got to be an impurity of some kind, or how do you know it's DNA and not RNA, or something else, or something going along, or that sort of, those were the questions at the time. So we needed more evidence. While Hershey and Chase did conclude that DNA was a heritable material, we didn't really know very much at all about DNA other than its basic chemical components. We didn't know about the base pairing. We didn't know about the anti-parallel structure. All we knew is that there were some nitrogenous bases, there was some sugar in there, and then there was a lot of phosphate in there. The reason I'm showing you this right now is that the phosphate's going to play a key role in the next experiment that really sealed the deal for DNA being the heritable material. So that was 1944. It's not another 8 years before we finally get the nail in the coffin experiment. There were lots of other experiments going on. These weren't just 2 or 3 experiments. There were literally dozens and dozens of people, hundreds of people around the world working on this question. But Hershey and Martha Chase really put the nail in the coffin for DNA as the genetic material. So we're going to go through this experiment, the Hershey-Chase experiment. Their model system involved bacteria and a viral molecule known as T2, the phage. It's a bacteriophage virus. So it's a virus specific for bacteria, and it's T2, a phage. What they did was they were able to selectively label the molecules in the phage, either the DNA or the protein. They know that the T2 phage is only made of DNA and protein. There isn't much else in there, not to a significant degree. And they could selectively label one or the other with radioactive isotopes. And then what they would do is they would incubate those labeled phage with E. coli to see if there was a transformation. Based on the radioactive data, they found that the DNA was the one that is the heritable material. So it was the nail in the coffin. There were several other nails that we haven't talked about, but this was the one that really sealed the deal. So here is the T2 bacteriophage that we were talking about that the Hershey Chase experiment utilized. Really, really cool structure. We know that it is only made of protein and nucleic acid. But awesome lunar lander looking kind of a molecule. I think it's one of the coolest molecules in all of biology. But we knew that there was only protein and only nucleic acid. And so Hershey Chase exploited that, being able to selectively label one or the other. So here are the details of the Hershey Chase experiment. What they did to begin was they selectively labeled either the proteins of the phage or the nucleic acids of the phage. Proteins have lots of sulfur in them, so if you grow up the phage over time in a culture with S35, the proteins will become labeled, while the nucleic acids won't. There is no sulfur in nucleic acid. So growing it up in conditions containing nutrient media in S35, eventually you'll get enriched radioactive viruses where their proteins have S35 in them. On the other hand, in a parallel experiment, they grew up the phage with radioactive P32. Now, proteins get phosphorylated, so there may be a little bit of radiation in there, but nothing compared to the amount of phosphate that you see in DNA. You have those phosphate backbones. So the phosphate backbones are going to have a lot more radioactivity than the proteins will. So you're relatively selective for nucleic acids when you grow up phage in P32. Then they took those radiolabeled phages. They grew them up in non-radioactive bacteria. So they grew up, they added the radioactive phages to non-radioactive bacteria. Once they let them grow for a period of time, they will infect those bacterial cells. And what they did was they then busted up the cultures, they blended them up, and they looked for where the radioactivity was found. When you lyse up the cells and you spin them, anything that was not injected into the cells would be up in what we call the supernatant. Anything that was injected into the cells would settle in the sediment containing the debris and contents of the cells. So the proteins of the phage would not settle, they would stay in the supernatant. And what we found was that the radioactivity, if introduced to this experiment, only happened when we used radioactive phosphorus. If we used radioactive sulfur, very little radioactivity made it into the cellular pellet. Only the radioactive phosphate settled into the pellet to any significant degree. And so that meant that what was being injected and transforming the bacteria was in fact a nucleic acid, because here none of the protein from those phages was making it into the pellet and settling into the bacterial pellet when you spun it down. Only when you had the radioactive nucleic acids did you see in the transformed bacteria did you see radioactivity in the pellet. So that was the nail in the coffin. And again, here's that really cool image of these bacteriophages looking like lunar landers. So we know now, in fact, you can even see here, these viruses are actually like DNA syringes. They are actually injecting physically the DNA into the bacterial cell. Other viruses, by the way, actually will merge with the plasma membrane of a cell that they're infecting. But in this case, the phage is physically injecting its DNA into the bacteria. All was going on with identifying DNA as a heritable material, a parallel effort was going on to understand the biomolecule structure in cells, and that included DNA. We were trying to understand what the structure was. They knew coming into Hershey and Chase that that DNA was polymeric and repetitive to the point that they thought it was just biological junk. It had nitrogenous bases, sugars, and phosphates, but that was pretty much it. A chemist by the name of Erwin Chargaff, though, noticed in observing DNA from different species that the composition from one species to the next varied. But he did also note some similarities across all of those species. So he was able to note the differences. There were slight compositional differerences, different ratios of nitrogen bases, different kinds of nitrogen bases in different species. But they all obeyed similar characteristics. So he saw differences and similarities. Those similarities became known as Chargaff's rules. So if anyone was going to come up with a DNA structure, that had to be able to satisfy what we knew about DNA from one species to the next. And so that's where Chargaff's rules came from. We knew about the diversity now from Chargaff, and we also knew about the similarity across the species. So Chargaff's rules, two major findings. That the composition of DNA does vary between species, and when we mean by composition, we mean the nitrogenous bases. You didn't have an equal amount of A and T and G and C in different species. However, even with that difference, the A's and T composition always were the same. You always had the same amount of A's and T's, and you always had the same amount of G's and C's. So the percentages of A and T were always identical, and the percentages of G and C were always identical. So we knew that there was variance across species, but they never violated this A and T and G and C rule. So when Watson and Crick were doing their modeling, they had to come up with a structure that could answer the reason for variance in rule number 1, but also answer the reason why A always equaled T and G always equaled C. It wasn't until we actually got to the double helix and people pretty much went, oh my goodness, how is it not so obvious to us? That's one of the beauties of a really profound finding in science, is that oftentimes it's deceptively simple. When people saw Watson and Crick's proposal, they basically slapped themselves on the head going, why didn't we think of that? Let's look at one strand of DNA as we now understand it. One strand of DNA. Now, it's pretty obvious to see that this one strand is asymmetric. We have this left side and we have this right side, right? In this picture anyway. We have this backbone, if you will, of alternating phosphates and sugars. So we're asymmetric from one side to the other this way, but we're also asymmetric in the vertical plane. This side is different from this side. We have the phosphates attached to the 5' sugar, and if you remember back in the day when we have the oxygen here in this circular sugar, we go clockwise with the carbons. 1 or 1', 2, 2', 3, 3', 4', and then the 5th carbon, 5'. And so we have the 5' phosphate, and then downstream to the next phosphate we hook up to the 3'. So this end is known as the 5' end. It's kind of like in proteins. You know, you have the amino end and you have the carboxylic acid end. It's the same thing in DNA. It's asymmetric from top to bottom here. The 5' end and the 3' end, because the next phosphate that's attached here would attach to that 3' sugar. And here we have our nitrogenous bases, and we've talked about those before. Rings of carbon and nitrogen that comprise pyrimidines and purines. Pyrimidines are the smaller of the two, purines are the bigger of the two. Now, Watson and Crick were not working alone. They were not working alone. They were using evidence from other scientists in order to come up with their model. They actually didn't do any experiments in their paper. They just proposed a structure. They had some understanding from Chargaff, and they had some other understanding from some other scientists, but the story of how they really came up with DNA is actually a little bit of cloak and dagger, and it's one of the great, I would say, misfortunes in science for the people involved. So we knew that DNA was accepted as a genetic material by the time around in the mid 40s. We didn't understand its structure. Well, a laboratory headed up by Maurice Wilkins with Rosalind Franklin, and again, this is one of the great crimes, I think, in science, and I'll explain why. They were using a technique called X-ray crystallography. So they were taking molecules, they were crystallizing them. They were pure molecules of DNA or protein and so forth, and they'd put them in a chamber and bombard it with X-rays. And when X-rays would pass through the molecules, they would ricochet off of the atoms in that molecule. And crystals are very, very regimental in their structure. They have, especially in polymers, they have repeating structures. And so when you look at that refraction pattern, you can understand the arrangement of atoms in that crystal. So Franklin, who was working in the Wilkins lab, was the one who she was the one who used crystallized DNA and came up with the image that Watson saw and then conveyed to Crick, and then led them to make the decision about the structure that they would propose. Watson and Crick got the Nobel Prize, but Franklin surely deserved it, along maybe with Wilkins as well. But Franklin was the one who came up with the data that sealed the deal for Watson and Crick, and this was her image. What it showed was, and it's hard for us to interpret here, what it showed was that there was a sort of a helical structure going on. That as you shone X-rays through the DNA crystal, it created this helical pattern. It not only gave us a sense of the arrangement of the atoms, but also gave us a sense of the width of the DNA molecule. So they deduced from this picture that it was a helical structure. They also were able to determine the width, because you can figure out the angles that are involved based on where the X-ray is coming from. They could see the spacing. They didn't see variance in the width. If it was purine and purine binding, those big giant nucleic acids, they would have wide areas. And if it was pyrimidine and pyrimidine, it would have narrow areas. And let's take a look at some of this. So it's hard to see in this picture. Lastly, though, you could divine that there were two strands there. So they were able to see that it was a helical structure of some kind. They knew how wide it was, and they knew that it was two strands. Because if you play with the molecules, you can see that there's more than one kind of DNA that can be created. But the fact that the DNA makes sense not only from the data that we see in the X-ray crystallography, but it satisfies Chargaff's rules, and we'll talk about that a little bit later. But this is so celebrated with that we now see it in our media, in our arts. For instance, if you see the divergent series, you see you can see the little subliminal messages about DNA and eugenics. Or in Gattaca, they have an apartment that has this double-stranded DNA in the staircase. And so we see this showing up in modern movies and film and so forth. It's how celebrated it is. It's not just a scientific, but also artistic. If purines and purines were bonding together, it would cause bulges. So we knew from the refraction pattern that it was double-stranded, so it got rid of some of the 3- and 4-strand possibilities. But how to account for the bonding? Well, they knew that the shape was regular, so it couldn't be purines binding with purines. It couldn't be pyrimidines binding with pyrimidines, or you would have seen a bulging pattern. So what we know now is that we have one strand, and here's the 5' end running this way. Here is the 5' end on the other. We call this configuration anti-parallel. They are parallel, but they are opposed in terms of their 5' and 3' ends. And in order to satisfy Chargaff's rules, we knew that all A's had to have the same amount as T's. All G's had to have the same amount of C's. And so this bonding, along with the regular spacing, sealed the deal for the ATGCs that we now take for granted. So here's a space-filling molecule or model of DNA showing you the atomic radii, and you can see it's very, very regular. It's repetitive, but it allows for diversity inside the molecule itself. The information is literally protected in the interior of the molecule. But that 2-stranded also set us up for a copying mechanism that wouldn't destroy the DNA molecule in the process. And that was all just as important. How do you convey new information without destroying the information from before? Watson and Crick were trying all different kinds of different structures. This was one of their structures that they proposed, and you can see that waggle, if you will, of width a little bit here. It's a little bit tough to see, I guess. They originally thought it was going to be A with A and so forth, but A is a purine, so purine and purines would cause this varying width, but we didn't see that in the X-ray data. What they concluded, because it was regular, and of a certain width, when they measured the length of the bonds and so forth, it was consistent with the X-ray data that it was a purine with a pyrimidine. So too wide for purines and purines, too narrow for pyrimidines and pyrimidines, but this would also satisfy Chargaff's rules. Because of the structures of A and T, they also determined that only 2 hydrogen bonds can form. There's no way for hydrogen to bond in this third arm right here. So A and T bind together with 2 hydrogen bonds. And G and C, because of the structure of the nucleotides, bind with 3. That 2 versus 3 actually becomes incredibly important when you are working with DNA and RNA in the lab, because 3 bonds is stronger than 2. Just like 3 people pulling on a rope is going to give you more force than 2. And that actually affects the properties of double-stranded and single-stranded DNA when you're binding it to the other strand. Make sure you have a good familiarity with the basic structure of the DNA double-stranded molecule. Phosphate backbone, where the phosphates go 5', 3', 5', 3', 5', 3'. Anti-parallel. A's double hydrogen bond with T's, and G's double hydrogen bond with C's. So make sure you have that structure down, understand the evidence, and that led to us not only as DNA as a heritable material, but the basic structure of DNA. Okay, we're going to stop right there for now. So this is the end of Part A, and I will see you in Part B. Thanks for your time. Please let me know if you have any questions, anything I can help you with that is not making sense. See you soon.